Hello. The students who have joined, can you please write in the chat box if my voice is clear and the presentation is visible? Please write in the, okay, thank you so much. We will wait for a couple of minutes till more students join, and then we will start today's session. Okay, so let's start today's session. Eventually, when students will join, we will add them. Very good evening to the students who have joined today's session. My name is Moshumi Dev, and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad, and I am the TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is the week 12 live session for this course, and this is the last live session which we will have for this course, and today we will discuss about the week 12 course content and some 10 to 15 questions related to that course content. So, if during the live session if you have any doubt, any query, please do write in the chat box. At the end of the session I will answer those queries, and if you cannot hear my voice or see the presentation, please also write in the chat box. Okay. So, now we will start today's session. So, the first question, it says that the DNA synthesis checkpoint is present in dash phase of the cell cycle, and the options are as follows: option A M, option B S, option C G1, and option D G2. So, Actually, when the cell cycle is something is happens when the cell is getting divided. Okay, so we all know that for the growth of the human body, the cells are continuously dividing and the cell death are also happening. So in that place, new cells have to form. So one cell is divided into two cells and the two cells divided into four, and by that way the cell division is happening. So in the cell division, the cell cycle is a major point of the cell division. So in this cycle, the circular points of some reactions happening, and the cell is getting prepared and then it is getting divided. So in this phase, one point is there is a DNA synthesis, and the DNA synthesis happens at a particular point of the cell phase, and there is so you have to identify now at which checkpoint the DNA synthesis checkpoint is present in which phase of the cell cycle. We will first see the answer for this question, and then we will discuss more about the cell cycle and the various checkpoints present in the cell cycle. So the correct answer for this problem is number B, S. So in the S phase of the cell cycle, the DNA synthesis checkpoint is present. Now this is what is called is the cell cycle and the various checkpoints present in the cell cycle. So as you can see, the first is the cell growth checkpoint. Now what is the growth phase? Is the G1 phase. It is called the growth phase, which is the first phase of the cell cycle. So here there is a checkpoint which is present, which is called cell growth checkpoint. And what happens here? It occurs toward the end of this growth phase. Here it is present, the cell growth checkpoint, and what it does, it checks whether the cell is big enough and has made the proper proteins for the synthesis phase. And if not, then the cell will go to this resting phase of G, which is called the G0, which is the resting phase, until it is ready to divide. Now after the G1 or growth phase, then we will have something which is called a synthesis or S phase. In this S or synthesis phase, the DNA synthesis checkpoint is present, and this phase what it happens, this checkpoint checks that whether the DNA has been replicated correctly, and if it is replicated correctly, then the cell will proceed to the mitosis phase. And there is also one phase which is called G2 phase, which is basically similar like G1, where all the checkings happens and see if the cell is ready for the cell division. And what happens in the mitosis phase? In the mitosis phase, the mitosis is complete, means there is various cell division like prophase, anaphase, metaphase, which will we have in this mitosis or M phase. So here the cell division is basically happening. So in all these phases, the cell is getting prepared for the division, and this M phase, the cell division is happening. Okay. Now we all know that what is cancer? Cancer is something which is called the abnormal growth of the cells. And why the cancer happen? Basically the cancer can result when one or more of these checkpoints malfunction. Means if sometimes it can happen that the cell is not ready, but for the synthesis, but till it goes to the G1 phase to S phase without going to the G0 phase. So by this mechanism the cancer can happen, and there is a various reasons for which the cancers can happen. So the cell cycle, it is very important for your course and for your knowledge also. So you please remember what are the different phases of the cell cycle, what are the different checkpoints are present, like there is cell growth checkpoint, DNA synthesis checkpoint and mitosis checkpoint. Where these checkpoints are present, like the first one is present in at the end of the G1, the second one in the S phase, and the third one is the start of the M phase. Okay. And what is the role of each and every checkpoint, and what the malfunction can do. So you have to remember these points from this topic. Okay. So I think it is clear what is the cell cycle, and this question itself is clear. And if you have any doubt, please write in the chat box. Otherwise, I will move toward our next problem. Okay. So let us move toward our next problem. The problem number 2, it says that palbociclib is the inhibitor of dash kinase. So you have the options are JAK kinase, option B BCR-ABL kinase, option C CDK4/6 kinase, and D MET kinase. So you have to identify this about this drug palbociclib. It is an inhibitor of which kind of kinase. Okay. So actually the kinases are one kind of enzyme which are very important enzyme, which basically transfers one phosphate from the ATP to a basically to a protein or lipid. So these are the there are several families of kinases in our body. So this Jack kinase, BCR, ABL, CDK, MET kinase, these are the several examples of the kinase in kinases. Okay, and this kinase also have a very important role in our body and the cell growth and cell cycles. So this is a drug which is called the palbociclib, which is an inhibitor of a particular kinase, and you have to identify for which kinase this drug acts as an inhibitor. So I will first show you the answer, then we will discuss more about this kinase and the inhibitors. So the correct answer for this question is option C, CDK 4/6. For this family of kinase, this palbociclib act as an inhibitor. So what is CDK 4/6 kinase? Actually, this called the cyclin dependent kinase. The CDK stand for cyclin dependent protein kinase. So it is nothing but it helps in the cell cycle. So basically this kinase enzymes, they work during the S phase of the cell mechanism. Okay. So all we as previous slide we also discussed the cell cycle. So where it happens like in the first G1 phase, the cell accumulates resources for the needs to duplicate, which are needed to duplicate the genome, and if it is not sufficient, it will go to the G0 in the resting state. And if it is okay, somebody wants to join. Okay. So like in the previous slide we discussed about the cell cycle and how in the G1 phase the cell accumulates the resources for which are needed to duplicate the genome, and if it is not there present at the end of the G1 phase, then the first checkpoint will send the cell to the G0 resting state. Then if it is ready enough, then it will be sent to the S phase or the synthesis phase, where the actual duplication of the genome occurs, means the DNA synthesis occurs. And if it is ready enough, then it will be go to the G2 state, where the proper DNA replication is checked and final preparations for the mitosis M phase is happening. And at the end of the cell cycle, the M phase, the chromosomes are separated and actual the cell division is happening. So what this CDK kinases are doing, they are basically helping during the this various phases of the cell cycle. And there is actually when there is a malfunction, like cancer and all, what happens like this during the malfunction and during the cancers, what happens that this kinases are over expressed. Okay. So when the overexpression of this kind of kinases are happening, the cells will be doing the malfunction and the G0 phase will be absent, and we will have what is called the malignant cells. So these malignant cells, they may not be have the proper functions of the normal cells. So that is called the actually the cancers. So these kinases are very important during the cell cycles, and which makes this kinase, particular this family of CDK kinase, CDK 4/6 kinase, and all these kinases can be targeted as a drug target for the treatment of various kind of cancers. So such a drug target such a drug is a palbociclib, which is a kinase inhibitor for this CDK 4/6 kinase family. Okay, so this is the structure of the palbociclib kinase inhibitor, and this is the this Ibrance is, it is a commercial name for this drug. So basically it is a type 1 kinase inhibitor. So there are various kind of kinase inhibitor, you don't need to know that at this point, but for your just knowing, I am saying that there are various type of kinase inhibitors depending on where these inhibitors are binding in the kinase enzyme. So if it is binding in the ATP binding pocket, it is called the type 1, and it is if it is binding covalently, it is called type 6. So there are different kinds of type of kinase inhibitors. So this palbociclib, it is a type 1 kinase inhibitor of CDK 4/6, and it binds to the ATP pocket in an active conformation through 2 hydrogen bonds with the kinase hinge valine 101 and one with aspartic acid 163 of the DFG motif, and its cyclopentyl group occupies the ribose pocket. As you can see, there is a cyclopentyl group. So it is used for the treatment of hormone receptor HR positive, HER2 negative advanced or metastatic breast cancer in post menopausal women, and there is side effects are neutropenia and leukopenia. So you don't need to remember each and everything like how it is binds or no, but please remember this name of this drug, because CDK kinases are very important and for this cancer treatment, I will suggest you, please remember one or two names for the each and every class of the kinase inhibitor. You can read it from any books or any journals, or you can find it in Google. It is with the kinase inhibitors are very extensively used nowadays for the treatment of the cancers, and you will find very good reviews for regarding that. So I will suggest please remember one or two names for the drugs, because it is very important, and if you know the mechanism how it is happening, then it is beneficial. Okay. So I think it is clear this problem number 2 about the palbociclib and the CDK4/6 kinases. And I am saying please, if you have any doubt, write in the chat box. At the end of the session I will try to answer those queries, and if you have any query and if you cannot hear my voice or see the presentation, please also write in the chat box. Now we will move toward our next problem. So problem number 3, it says that which enzyme is involved in the activation of the prodrug cyclophosphamide. So the options are number A, cytochrome P456, so 450, so option number B is the MMO, option C cytochrome C oxidase, and option D is the SOD, which is the superoxide dismutase. So you have to identify the correct enzyme which is involved in the activation for the prodrug of the cyclophosphamide. Okay. So now I will show you the answer first, then we will discuss about the cyclophosphamide. So the correct answer of this question is the number A, cytochrome P450. So this enzyme is involved in the activation of the prodrug of the cyclophosphamide. So what is the mechanism of action? So the cyclophosphamide, it is you can see the structures from this slide. This is the structure of the prodrug of the cyclophosphamide. So this cyclophosphamide is most commonly used alkylating agent, and both cyclophosphamide and ifosfamide they are first biotransferred to the hydroxylated intermediate primary in the liver by the cytochrome P450 system, and the hydroxylated intermediate then undergo breakdown to form the active compounds phosphamide mustard and acrolein. Reaction of the phosphamide mustard with the DNA is considered to be cytotoxic step. Okay. So this is the overall mechanism for this activation of cyclophosphamide, and as you can see, the first step, it is the activation of this prodrug to the hydroxy form by the cytochrome P450 enzymes. So what happens that this hydroxylation happens at the C alpha position for next means the carbon next to the NH group, and after the ring opening and all the mechanism, we have two reactive species. One, we have this acrolein, which is considered as toxic, and another we have this phosphamide musters, which forms this kind of intermediate reactive species, and this reactive species, it reacts with DNA, and this is the cytotoxic step. Okay. So I will suggest if you don't need to remember this whole mechanism for this, but remember how it is happening, and the name of the cyclophosphamide itself, it is very important alkylating agents, and if you can remember the mechanism, it is very good. Now, so please remember the name of this enzyme, which is the cytochrome P450 enzyme, and this is all about the cyclophosphamides, and I think it is clear. I will move toward our problem number 4. Okay. So yeah. So what is our next problem? Let's move toward it. So problem number 4, it says that identify the incorrect statement about mitomycin C. So the options are as follows. Number A, it contains an aziridine ring. Number B, it is not a prodrug. Number C, the first step in its method of action is reduction by NADPH, and option D, it forms a indole-hydroquinone intermediate. So this mitomycin C, it is a one kind of a drug actually. So you have to identify the correct statement about this mitomycin C, and let's see the answer first, then we will discuss about this mitomycin and the mechanism of its action. So the correct answer is the number B, it is not a prodrug. So this is the structure for the mitomycin C. As you can see, it is actually I will just okay. So for the mitomycin C, there is a aziridine ring present in the mechanism. As you can see, this is the aziridine ring. I will just mark it, so it will be easier for you. So this is the aziridine ring which is present in the mitomycin. Okay, this is a 3-member ring which have a nitrogen. It is called the aziridine, so the first option, it is correct about this mitomycin C because it has a iamidine. So it is not a prodrug. It is a wrong answer. It is a prodrug. Okay, so this mitomycin, like the cyclophosphamide, it is a prodrug, and it needs to get activated, and the activation which happens by the NADPH or cytochrome P450 reductase, and this enzymes, it happens, it helps for the reduction of this quinone ring. As you can see in the mitomycin, there is a quinone ring present. This is a quinone ring of the mitomycin. Okay, so at the first step, for the activation, this quinone, it is getting getting reduced to the hydroquinone, which is called the mitomycin hydroquinone, and after this, there is a action of methanol, and this after the reaction of this methanol, what will happen, there will be a incorporation, there will be elimination of this methanol from this ring, and we will have this kind of double bond here. So here we have a hydrogen, and if you eliminate the methanol from this step, we will have a double bond here, which is this, and this structure, it is called the indole-hydroquinone structure. Okay, so as we know about the structure of the indole, what is the structure of indole, we all know, and I will just draw it. So the structure of the indole is like this. There is a 6-member and there is a 5-member ring like this. So this is the structure of our indole, and the indole-hydroquinone is nothing but there will be 2 OH group at this para position of this benzene ring of the indole. So this is the basically indole-hydroquinone intermediate for this mitomycin C, and after the reaction of DNA, it forms the mitomycin DNA adapt cross-linked DNA, and this is basically there is a Fenton reaction happens here, by which this reaction is happening. So this is the incorrect statement, and this is also correct, and this is also correct. Okay, so I will please remember about the mitomycin C. It is a very important drug, and the mechanism of action, if you can remember, I will suggest please remember the mechanism of action of mitomycin also, because it is very important, and the structure, if you can remember, it is good. So I think it is clear. If you have any doubt, I am again saying, please write in the chat box, otherwise I will move toward our next problem. So the problem number 5, it says that which of the enzyme is involved, which enzyme is involved in the method of action of okay, sorry, there is a typographical error in this question. It will be which reaction, it will be reaction, not enzyme. I will just correct it. It is not the enzyme, it will be reaction. So the question will be which reaction is involved in the method of action of the calciamine. Okay, so this is a drug basically, and there is a one mechanism, the one name reaction which is involved in this method of action of this drug. So first I will say you the answer, then I will just correct it, and then we will discuss it. So the correct answer for this problem is the Bergman cyclization. The options were option A, Bailey-Silman reaction, Suzuki coupling, Diels-Alder reaction, and Bergman cyclization, and the correct answer is option D, Bergman cyclization. I will just correct it here itself. Okay, so now it is corrected. So the correct answer for this reaction is this question is the Bergman cyclization. So this reaction is involved in the method of action of this calciamine, and this is a one kind of drug. So what is the Bergman cyclization? So it is basically a radical reaction, and the starting material for this reaction is the endiene. So what is the endiene? Basically it is a triple bond followed by a double bond, and then there is another triple bond. So there is a in, which is and there is a diene. So this in, it stands for the double bond, this in stands for the double bond, and this ine stands for the triple bond. So after the reaction with the heat or H nu, there will be a formation of a dry radical here, which is a 1,4 benzene diol diradical. or parabenzene benzene 1,4-benzenoid or parabenzene diradical, and after the reaction what happens, this kind of cyclization happens, which gives us a benzene ring and a benzene derivative. Okay, so this Bergman cyclization is takes part in the method of action of this drug, and I will show you how. So this is the drug which is called the calciamycin, and this is what happens, there is a disulfur bond here present. So at the first step there is the elimination of this nucleophile, it takes part and reaction, and it breaks this disulfide bond, and we have a thiol here. And what happens, this thiol will be reacting, actually there is a mechanism, radical mechanism kind of mechanism, and this not radical mechanism, this thiol will be acting as a nucleophile, and it attacks to this double bond, and this actually this alpha beta unsaturated ketone kind of group, and then there is a, somebody wants to join. Then what happens, like after the thiol attacks to this alpha beta unsaturated, there is a resonance kind of there is a resonance here, and then what will happen, there is a, as you can see, there is a end-diene formation here also. As if you carefully see, there is a end-diene formation, there is here, it is present, there is a triple bond followed by a single double bond, and then another, there is a single bond, and there is a triple bond. So it is a end-diene kind of formation like this, and there is a, then there will be a Bergman cyclization, and we will have this 1,4-benzenoid diradical, and what happens, this DNA it will attack the DNA, and DNA diradical will form, and DNA it will cleave the DNA double strand. Okay, so this is the mechanism of action. This may be a little difficult because of the structure of this particular drug, so you don't need to remember the exact mechanism, but actually in your exams this mechanism can be asked. Okay, so I will suggest, if you can remember, it will be helpful. So basically the mechanism is not itself very difficult, it is the structure, it may seem very complicated, but if you practice, it is very easy. And remember the name of the cyclization which is happening in this method of action of this particular drug. So now I will move toward our next problem. So the problem number 6, it says that the method of action of aromatase inhibitor is option A, irreversible inhibition, option B, reversible inhibition, option C, suicide inhibition, and option D, uncompetitive inhibition. So aromatase, it is a important enzyme of our body, and this aromatase inhibitor, which basically inhibits this particular aromatase enzyme, and we will see in short why this is needed, why we have to inhibit this aromatase enzyme if it is important to our body. And like kinase inhibitors, aromatase inhibitors are very important, one of a very important class of the drugs. So see, there are many much enzymes in our body which are very important for particular biotransformation and biological reactions, but when there is a overexpression of this kind of enzyme, so or in a particular disease, so then we have to inhibit this kind of enzyme. So and it is a very this inhibitor of the enzyme inhibitors are very important drug targets nowadays for the for various kind of diseases. So let us see the answer first. So the correct answer is the suicide inhibition. So the method of action for this aromatase inhibitor is the suicide inhibition. So now I had said that aromatase is a very important enzyme which basically catalyzes the reaction of the transformation of the aldosterone and testosterone to the estrone and estradiol. And why it is needed, like if this estrone and estradiol, they are generated in excessive amount or in the malfunctions cells, what will happen, there is a ER activation and by this estrone, and generation of E2, 3, 4 quinone by this estradiol. And this first one, it will basically helping in the proliferation of the mutated cells, and this second one, it will catalyze the DNA alkylation and depurination, and which will basically results in the cancer. Okay. So the aromatase inhibitor, it stops this aromatase enzyme, and this this reaction cycle will be break in the first step itself. So this cancer does not happen. and this aromatase inhibitors are very important class of the drugs, and this is the mechanism of action. You don't need to remember it, I just has given it for your just knowledge. So, as you can see, the active site, it contains iron 2, and there is a iron porphyrin system here present, and at the distal side there is a sulfur, cysteine sulfur present with this iron, and after at the first step the oxygen, it is basically oxidizes this iron 2 to iron 3, and at the like hemoglobin, the iron is present in the top of this porphyrin ring, and after the oxidation it fits in the porphyrin ring center, and and then there is a electron donation, basically there will be O minus generation, and this cycle happens. Okay. So, I give this cell cycles because you can be asked about this aromatase enzymes, like which is the metal ion present in the active site. So, you have to remember that there is the iron 2 which is present in the active site, and it is a iron porphyrin system. So, as so because of so if you can maybe ask this type of question, that is why I given this cycle. If you cannot remember it, it is okay, fine, but if you do not remember the mechanism itself, but if you can remember this particular structure, it is very good, I will say. Okay. So, I think please remember how this what is the role of this aromatase enzymes, and the method of action is the suicide inhibition. Okay, and we discussed in the suicide inhibition in our previous lectures also. So, you can reread it from there. I am not discussing it right now, we will just move toward our next problem. Okay. But if you have any doubt regarding it, you can write in the chat box, or you can just mail me, I will then answer your query. Okay. So, problem number 7, it says that which of the following is not involved in the treatment of the PUD. So, options, this PUD, it stands for the peptic ulcer disease, and I wrote the abbreviation because you may be in the exam, you can be asked about it is like itself is a PUD. So, if you remember the abbreviation, it is very good. Okay. Peptic ulcer disease. So, it is the full form of the PUD. So, this is basically nothing but all you know in the layman's term, it is the acidity or the which problems happens in our stomach. Okay, the excessive acid formation in the stomach which results in this peptic ulcer disease, or we are very familiar with this kind of disease and drugs. So, the options are as follows: option A, omeprazole; option B, ranitidine; option C, misoprostol, and option D, levocetirizine. So, you have to identify the correct drug which is not involved in the treatment of the PUD. Okay. So, let us see the answer first, then we will discuss it. So, the correct answer is number D, levocetirizine. Actually, levocetirizine, it is not a drug for the peptic ulcer. Levocetirizine basically is used as a in the allergies. When you have allergies, then the and you are sneezing all the days and the whole day, then doctor prescribes this levocetirizine, which is basically the acts on the histamine, which is produced in excess when we have any allergy, seasonal diseases. So, the first three, these are used in the PUD or the peptic ulcer disease. So, the drugs for the peptic ulcer can be divided in the following classifications. Okay. So, there is there are the following drugs. So, there is a gastric acid secretion inhibitors, there is gastric acid neutralizers, or which are called the antacids, and there is ulcer protectives, and there is anti H. pylori drugs. So, this anti H. pylori, this H. pylori is a bacteria which helps in the actually the acid formation, and these all are so there are examples of the drugs like the gastric acid secretion inhibitors are of 4 types. This is like H2 antihistamines. There are proton pump inhibitors, there are anticholinergics, there are prostaglandin analogues, and the gastric acid neutralizers or antacids are like systematic antacids and non-systematic antacids, and there are ulcer protectives like sucralfate and colloidal bismuth citrate, and anti H. pylori drugs are like antibacterial drugs we are know, like amoxicillins, there is a metronidazole, and these are the drugs. And there are several examples, like ranitidine is a very famous drug for this peptic ulcer. We all know, we have taken it at some point of our life, and it is a H2 antihistamines, like there is a pyrazapine, and there is omeprazole, lansoprazole. These are very common drugs. So we all, every time we have this kind of acidity type of problem, the doctor suggests this kind of drugs, and there is a misoprostol also. So I remember this examples for this peptic ulcer drugs, and this classification also is very important, as where this is a very this is very common. And so I will suggest remember the classifications at first, and remember one or two examples for each class, so it will be enough. So I think it is clear. Now I will move toward our next problem. So the problem number 8, it says that the calcium channel blockers are used for the option A, hypertension and heart diseases, option B, leukemia, option C, peptic ulcers, option D, type 2 diabetes. So calcium channel blockers are a class of a drug. So these are used for a particular diseases, and you have to identify the correct answer. So let us see the answer first, and then we will discuss what are the calcium channel blockers. So the correct answer is number A, hypertension and heart diseases. So the calcium channel blockers, these are the kind of drugs which are used for the heart disease and hypertensions. And the therapeutic uses of the calcium channels blockers are like angina pectoris. There is each and every examples for this class, like hypertension, like the hydropyridines, they are used for the hypertension, which are the calcium channel blocker. For migraine, the verapamil is used. For the stroke, nimodipine is used. So and there are also side effects for this this kind of drugs. So what is the mechanism of action of the calcium channel blockers? So they have 4 effects. So they act on the vascular smooth muscle and reduce the contraction of the arteries and cause vasodilation. They act on the cardiac muscles, the myocardium, and they reduce the force of the contraction of the heart, means the negative inotropic. So they slow down the conduction of electrical activity within the heart and slow down the heartbeat, the negative conotropic, and they block the calcium signal on the adrenaline cortex cells. They directly reduce the aldosterone production, which correlates with the lower blood pressure. We discussed that about the blood pressure, there is aldosterone, there is and there is ACE. These are related to the there is a renin-angiotensin system. So and this is directly linked to the adrenal cortex cells. So basically this calcium signals, the CCB, this blocks this calcium signal in the adrenaline cortex cell. So these are the 4 therapeutic effects for this calcium channel blockers, and I will suggest, please remember this. Basically these are very important, like this angina, for angina, cardiac failure, hypertension and stroke. For this this calcium channel blockers are very important. And remember 1 or 2 names for this calcium channel blocker. You can be given a name of a drug, and you can be asked which kind of drug it is. Is it a calcium channel blocker, or it is a proton pump inhibitor, or like this? You can be given the name of the drug, and you can be asked to write the therapeutic use for the therapeutic class for this particular drug. So I think it is clear. Now I will move toward our next problem. Okay, so problem number 9, it says that what is the correct order of the intermediates in the route of the cholesterol biosynthesis, and the options are as follows. Option A, HMG CoA to mevalonic acid to squalene. Option B, squalene to HMG CoA to 7-dehydrocholesterol. Option C, mevalonic acid to farnesyl diphosphate to squalene. And option it will be D, there is a typographical error. Option D, it is acetyl CoA to HMG CoA to squalene. So as we all know, cholesterol is a very important component of our body, and it is a very fancy term which is nowadays used for our diseases, because all you know that there is a higher cholesterol levels are not good for our body. And but actually what happens, like we all are said that we need to eat right, eat properly for so that the cholesterol generation is not happening in our body. But actually this cholesterol, the most of the cholesterol itself generated in our body, and there is a biosynthetic pathway, and it itself generates in our body. So, and this is a very important biosynthesis of one of a very important biosynthesis, and you have to identify the correct route of intermediates for this cholesterol biosynthesis. So, let us see the answer, then we will discuss the method. So, the correct answer is number C, the mevalonic acid, farnesyl diphosphate and the squalene. So, it is the correct order for this cholesterol biosynthesis. So, this is the overall chart for the cholesterol biosynthesis. It starts from our very known acetyl coenzyme A, and the first step this acetyl coenzyme A is converted to acetoacetyl coenzyme A, and the enzyme which is helping this conversion is the acetoacetyl CoA thiolase. And the second step, what happens, this acetoacetyl CoA with the with reacting with the acetyl one molecule of acetyl CoA, and it forms 3-hydroxy 3-methyl glutaraldehyde CoA, and the enzyme which is helping in this conversion is the HMG CoA synthesis. Okay, and this is basically the HMG CoA. This 3-hydroxy 3-methyl glutaraldehyde CoA, it is abbreviated as the HMG CoA. Okay, HMG CoA. And the third step, what happens? The fourth step also the third step, it happens that the HMG CoA is converted to the mevalonic acid, and the enzyme which is helping it, the HMG CoA reductase. Now, at this then what happens, this mevalonic acid converted to the farnesyl diphosphate, and this farnesyl diphosphate is converted to the squalene by the help of the squalene synthesis, and there are small steps in between this. So, ultimately this squalene is converted to 7-dehydrocholesterol, and which after in the help of the enzyme, this 7-dehydrocholesterol reductase, it is converting in the cholesterol. So, this is the overall biosynthetic pathway of the cholesterol biosynthesis, and I suggest you to remember this biosynthetic pathway. It is very important. You do not need to remember the structures for this this mevalonic acid or farnesyl diphosphate or squalene, but remembering the names and the name of the enzymes and which enzyme is catalyzing which step, it is very important. So, I will suggest, please remember this chart, this sequence, and remember the name of each and every enzyme and in which step it is helping. You have to remember this. Okay. I will just correct this typographical error here. Otherwise. So, it will be D. Okay. So, I think it is clear, and please as I said, please remember this chart, it is very important. Now, I will quickly move toward our next problem. So, the problem number 10, it says that statins work as option A, HMG CoA synthetase inhibitor, option B, HMG CoA reductase inhibitor, option C, squalene synthetase inhibitor, option D, acetoacetyl CoA thiolase inhibitor. So, as I said that the in our body the cholesterol biosynthesis is a very important reaction, but maximum cholesterol which is produced, it is produced in our body itself. So, the people we have higher level of cholesterol, they are given a particular drug which are very important, like statins, like atorvastatin. It is a very important and very well known cholesterol drug which are used extensively for the treatment of higher level of cholesterols, and this particular drug, it is following in the class of the statins, which are a drug, and this drug inhibit a particular step for this cholesterol biosynthesis. Okay, it inhibits a particular enzyme in the biosynthesis path of the cholesterol, and you have to identify which enzyme it is inhibiting. Okay. So, let us see the answer first, then we will discuss about the statins more. So, the correct answer is option B, HMG CoA reductase inhibitor. Like in our previous, we said that this is the HMG CoA reductase. So, what is HMG CoA reductase is doing? It is basically converting HMG CoA to mevalonic acid, and it is a very important step for the cholesterol biosynthesis. Now, statins are the class of the drugs which inhibits this HMG CoA reductase and inhibiting the cholesterol biosynthesis. Okay. So, So what is the HMG CoA reductase inhibitors or statins? The mechanism of action, they are analog of HMG CoA. They are competitive inhibitor of the HMG CoA reductase and the rate limiting step in the cholesterol synthesis, reduced hepatic cholesterol levels in the low dense LDL receptor expression, which the removal of LDL from the body, and reduced hepatic cholesterol results in the decrease in the VLDL synthesis and the secretion. So what are the adverse effects? There are adverse effects for these drugs also, which is the which is the hepatotoxicity in the less than 1% of patient it happens. The myopathy, which is 2 to 10% of patient, which rarely leads to the rhabdomyolysis, and the contraindicated in the pregnancy. So this is there are the few examples for the like atorvastatin. As I said, it is a very important, and I suggest please remember some few examples. Like atorvastatin is a example for this HMG CoA inhibitor, and I will suggest please remember few names for this statins, because they are very important class of the drug. And remember, like this point is, second point is very important, like they are the competitive inhibitor of the HMG CoA reductase, okay, and the rate limiting step in the cholesterol biosynthesis. So it is also this point is worth of mentioning here, like okay, like this step, this step of the conversion of the HMG CoA to mevalonic acid, it is the rate limiting step for this reaction, okay. So it is also worth of remembering. Also please remember this second point is itself is very important, and remember a few examples for the statins or the HMG CoA reductase inhibitors, because you can be given the name and you can be asked to say which kind of drug it is, okay. You can be name of this atorvastatin and you can be asked which kind, what is the role of this drug, okay. So I think it is clear how the statins work, and I will move toward our next problem. So problem number 11, it says that for the better bioavailability of a drug, what should be maximum number of the rotational bonds? And the options are as follows: option A 10, option B 7, option C 5, and option D 13. So bioavailability of the drug is a very important aspect of the drug, and in our previous lectures, in one lecture we discussed about this bioavailability of drugs, how the Lipinski's rule is working for the bioavailability of drug. So this number of rotational bond, it can be also suggested as one kind of part of this Lipinski's rule, and which detects the bioavailability. And there is a certain number for this rotational bond which cannot be exceeded, otherwise the bioavailability of the drug will be poor. So let us see the answer first. The correct answer is B. So maximum number of the rotational bonds should be 7. So as you can see, there is a I will say so 7 is the limit, okay, the limit is the 7. So maximum number of 7 rotational bonds is okay. Otherwise, if the number of bonds are more than 7, then what will happen, like the bioavailability of the drug will be poor. So what is the what is the rotational bond? So basically, as you can, this is a very good example. So a rotational bond defined as any single non-ring bond attached to a non-terminal non-hydrogen atom. Amide CN bonds are not counted because of the higher barrier of rotation. So as you know, as you can see from these examples, like these bonds, like this bond, this bond, this bond, this one, this one, this one, this one, and this one. So these bonds are the rotational bonds in this drug, and for this drug these bonds are the rotational bonds. So the definition of the rotational bond is very important, otherwise you will not know which are bonds are not rotable or which are not. So any single non-ring bond attached to a non-terminal non-hydrogen atom is called the rotational bond. But the amide, the normal CN bond is a rotational bond, but amide CN bond is not counted because of the higher barrier of the rotation, okay. Means this amide CN bond is not freely available to freely rotating. So this, there is an example, this is a very good example, like there are two drugs, one is the atenolol and is propanol. So these two drugs are in the first one the number of rotational bond is 8, and you can see the bioavailability for is 50% only. but for the second one the number of rotational bond is less, which is the 6, and we will have a very good viability of 90%. Okay. So, as you can see the maximum number is 7. So, the 6 it is below 7. So, this drug has a better viability, and when the number exceeds the number 7, means when the number of rotation bond is greater than 7, the viability decreases drastically by the towards the 50%. Okay. The number of rotation bond is a very important aspect for the drug viability, so remember this thing and this example also. So, I think it is clear, and I will now move to our next problem. Okay, so problem number 12, it says that which metal ion is present in the active site of cytochrome P450. The options are number A magnesium, option B nickel, option C zinc, and option D iron. So, cytochrome P450 is a very important class of the enzymes for our body, and we all know that it is a metalloenzymes actually, because in the active site there is a metal ion present, and it is very important for the functioning of this enzyme. That is why it is called a metalloenzyme. And in the whole course we said we had seen that in many drugs or basically the activation of prodrugs to the actual active form, it is catalyzed by the cytochrome P450 enzyme. So, it is really important to know what is the active site and what is basically the structure and the active site for this enzyme, and let us see the answer first. So, the correct answer is option D iron. So, iron is the present in the active site for this cytochrome P450 enzyme, and this is the heme iron. Okay, this is a heme. There is a iron and this is a heme enzyme. There is classified. So, what this kind? So, there is just one example. This cytochrome P450 it catalyzes many reactions. So, it is one kind of reaction. This is basically incorporation of oxygen. Like there is a SH bond, and this enzyme, which what it happens, it does, it is incorporating oxygen into this SH bond. Okay. So, there is actually you can see there is this is the active form. There is a iron 3, and this in the porphyrin ring, and there is a cysteine sulfur in the axial position, and after the reaction with this oxygen and NADPH, and this is the substrate thiol, what happens that this iron 3 it is attached with this oxygen, and there is a basically a peroxide kind of linkage happening, and by this there is a active form, which this is very this is the active intermediate, which is the iron 4, and this is a cation, radical cation. There is a positive charge and there is a radical, which is called a radical cation, and this radical cation it activates and reaction happens like this. Okay. So, remembering this mechanism, if you can this is out of your this is not directly this enzyme, this mechanism is not present by but I just thought it will be good for you to know about the active site mechanism for this very important enzyme, because in the most of the product to drug activation you have seen the cytochrome P450 enzyme is involved. So, this is the mechanism of action. If you don't remember, if you don't need to remember the exact mechanism, but if you can remember, there is the several things like there is iron 3 in the center, there is a axial position, there is a cysteine sulfur, and what it does, and this intermediate is very important. So, if you can remember this intermediate, I will suggest it is very good if you can remember this kind of intermediate. Okay. So, I think it is clear. Now, I will move to our next problem. So, the problem number 13, it says that selegiline is a specific inhibitor of option A myo B, option B myo C, myo A, option C ACHE, and which is the acetylcholine esterase, and option D ACE, which is an angiotensin converting enzyme. Okay. So, this selegiline is a very important inhibitor of one of these enzymes, and let us see first the answer, then we will discuss this. So, the correct answer is option A myo B. So, selegiline is a specific inhibitor of the myo B. So, this myo stands for monoamine oxidase myoamine oxidase. Okay. This myo stands for the myoamine oxidase, and they are of two types. There is a myoamine A and myoamine B enzymes, and as you can see here, so there are two kinds of myoenzymes. So, myo A enzyme is present in the cytoplasms of the neurons, the central nervous system, and peripheral tissues of the liver, like in the liver. It acts non-specifically on Na, 5-HT and dopamine, and chlorothalidine is a specific inhibitor of the MyoA. And MyoB, it is present mainly in the CNS and acts more on the dopamine, and seleginine is a specific inhibitor of the MyoB, and paraglidine is a non-selective MyoI, because it is not selective, it can inhibit MyoA or MyoB. So, this myoamine oxidase inhibitors are a class of powerful antidepressant drugs. They are particularly effective in treating social phobia, panic disorders, atypical depressions, and due to potentially lethal dietary and the drug interactions, myoids have been reserved to a last line of the RX, used only when the other classes of antidepressant like SSRIs and TCS have failed. Okay. So, this myoamine oxidase inhibitors are very important class of antidepressant, but due to potentially lethal dietary and drug interactions, they are reserved for the last stage of the treatment. So, there are many classes of antidepressant drug, like there is a some SSRI, there are TCS, and if these class of drugs are failed, then MyoI are used. Okay. So, remember what is the role of the myoenzymes, and this examples of one or two examples which will be enough. So, now I will move toward our next question. So, the problem number 14, it says that which of the following is not a QSR parameter? Option A, hydrophobicity, option B, hydrophilicity, option C, steric, and option D, electronic. So, what is the QSR? QSR is the quantitative structural activity relationship, which is one aspect of the drug discovery, and it is basically it falls in the computational drug discovery thing. So, there are various parameters by which this QSR are judged. So, you have to identify the correct parameter which is not a QSR parameter, which is the option B, hydrophilicity. So, what is a QSR? A quantitative structural activity relationship, which is and it is a mathematical relationship which correlates measurable or calculable molecular properties to some specific biological activity in terms of an equation. So, it is basically correlating the mathematical equation and our bodily reactions, biological activity in terms of an equation. Okay. So, this QSR attempts to find consistent relationship between biological activity and molecular properties, so that these rules can be used to evaluate the activity of new compounds. Like if you have a particular value of a particular parameter, you can say that if the drug is good or bad, or how much it need to be improved for a better reaction or better reactivity. Understanding QSR regarding electronic effects, steric effect, and lipophilicity. So, physiological physicochemical parameters, various parameters are used in QSR studies are hydrophobicity, like partition functions, pi substitution constants, these are the parameters. Now, for the steric parameters, we have Tufts equation or Varloop steric parameters, and electronic parameter we have Hammett constant and dipole moment. So, these are the various parameter and various constants which are various particular variables for this QSR. So, overall knowing this QSR, what is the QSR and what is the parameters are enough. You do not need to go details, and if you can remember few examples, few equations, it will be good. But knowing what is a QSR, understanding it is very a little bit of difficult, because it is a completely mathematical, and correlating it to basically computational chemists do this kind of QSR techniques in the drug discovery. And so, remember these three are very important. This three parameters, this hydrophobicity, this steric, and this electronics are very important parameters for the drug discovery of the molecule itself. And what are the parameters in this, each of this class, you have to remember also. Okay. So, I think it is clear. Now, I will move toward the last question of today's session. So, the problem number 15, it says that anti-ulcer drug omeprazole is option A, reversible proton pump inhibitor, option B, antagonist of H plus K plus ATPase, option C, agonist of H plus K plus ATPase, and option D, irreversible proton pump inhibitor. So, we in the previous slide we discussed about this peptic ulcer, and omeprazole is a very well known drug. We have taken it at any point of our life for this our acidity problem or ulcer problem or digestion problem. So, it is basically a particular class of this class of the drug, and you have to identify the correct answer. So, I will quickly show you the answer. Thus, the correct answer is number D, irreversible proton pump inhibitor. So, this is the structure of the omeprazole. Okay. So, as I always say, please remember few structure of very common drugs, like omeprazole is a very common drug, and you can be given this structure and you can be asked to write the name and which type of drug it is. Okay. Since it is a very common. So, for very common drugs, I will suggest you to, if you can remember the structure, it is very good. So, what are the proton pump inhibitor? The mechanism of action, as you can see, omeprazole, which is like Prilosec, and it is a commercial name, and lansoprazole, which is Prevacid, and they inhibit the proton pump effectively, irreversibly, and requiring synthesis of the new enzyme protein. So, omeprazole and lansoprazole approved for the treatment of duodenal ulcer, which may be in the conjugation of the triple therapy, erosive esophagitis, and gastric acid hypersecretory states, including Zillinger Ellison syndrome. Okay. So, this gastronomus that cause the secretion of the large amount of acid. So, basically proton pump inhibitors work by lowering the amount of the acid secretion in our stomach, so that the ulcer does not happen. So, they are very important class of this anti-ulcer drugs, and omeprazole is a very common and well known drug for this kind of drug. So, this was the last question for today's session, and this was the all about the week 12 for this course of the organic chemistry and biology and drug development. And today it was the last online session for your live problem solving session, and I will just share my email ID quickly in our in my chat box. So, this is my email ID which I have just shared in my chat box, and if you have any doubt regarding any course content about any week, you can feel free to contact. You can mail me, and you can also write in the discussion forum. And this was the last interaction session. In previous 12 weeks, we have discussed about various terms for this organic chemistry and biology in drug development, and this PPT and this class video, it will be shared to you in the NPTEL website. From there you can download today's about today's session and this slides, and if you have missed anything during the session in any in today's live session, you can also write in the you can write to me, and you can find it from there. And so, basically it all was about the organic chemistry and various in today's this week's session, we have various kinds of drug like anti-cancer drug, we had anti-cholesterol kind of drug, and the this acidity, and all we have discussed about this. And QSAR like is a very important aspect for the drug development, and also there was cholesterol biosynthesis, which I have says it is a very important topic for your exams also, or your knowing. And we discussed about the different kind of treatment of heart blocker, aromatase inhibitors, and cancer inhibitors, kinase inhibitors. So, that is all, and I thank you for joining today's class, and thank you for attending the students who have attending the classes regularly. And now I will just close today's session, and if you have any doubt, you can write in the chat box now. Thank you so much for the people who have joined regularly, and now I will close the meeting, and all the best for your exams who have the given for the annual final exams. So, all the best to you, and thank you so much.